Hello, everyone. It's a pleasure to present here to you and share some of my ideas about precision oncology and the whole promise of precision oncology and precision medicine in general also with you. I'm Istvan Petak and I'm founder of Genomate Health, but I also teach at various universities. I'm an adjunct professor at the University of Illinois at Chicago, also back in my home country in Hungary, Budapest, Semmelweis, and also a University of Jude, Szechenyi University. And I'm a physician scientist, so I trained as an MD. I worked in pediatric oncology at the beginning. I worked at St. Jude Children's Research Hospital for a while, but in the past 30 years, and now it's an important year for me. Actually, I started my career exactly 30 years ago. I've always been involved in various stages of what we call today the precision oncology, the very simple and logical concept to use our understanding of the tumor biology and the molecular pharmacology of targeted therapies to reason out, to choose the logical targeted therapy for every cancer patient based on the individual molecular profile of their tumor. So to highlight this dilemma, I usually start with a story. Just ask you to imagine that you meet a patient with a big nail in his head who complains about terrible headache. And as a doctor, what would you do? And of course, according to evidence-based medicine, we should treat based on the symptom and use the therapy that is statistically most likely to work. In this case, we should use any therapy against migraine, which is the most important cause of headache in most men. And so you should ignore your functional knowledge and understanding of how a nail in a head can cause pain for the patient. So it's a radical example, but it highlights the very basic idea behind precision medicine, which basically, in my terminology, means that to personalized treatment of patients based on the molecular level decision making, really understanding the molecular background of the tumor in every cancer patient and choosing the right therapy accordingly. So in my 30 years, I started in 95 before any targeted therapies were approved, and we didn't have the human genome ready. And then it's amazing journey to witness in these 3 decades how we sequenced the human genome, developed the first companion diagnostic tests, identify the first targets, imatinib in 2000, and trastuzumab even before in 97, and then gefitinib later. I will have some slides about that story, and until today. Now, the big question of this talk we try to answer: how far are we now? Are we there yet, or is still away? And is it going to take us 14.4 million years to really find the right targeted therapy for every cancer patient based on the individual molecular profile? Or can we do this faster and finish it in our lifetime? And in the audience, I'm sure there is a lot of scientists, professionals, biologists, laboratory scientists, and and physicians who are excited about the idea to use our understanding of the biology and laboratory test results to make individual treatment decisions for every patient. And basically, to summarize again the problem, I think William Osler, who was the founder of Johns Hopkins University, beautifully said that medicine is a science of uncertainty and art of probability. Really summarize well the challenge of medicine, where we always have to balance evidences and to make the right decision for our patient. Now, in the 90s, when I graduated from med school, was the high time or the prime time for what we call evidence-based medicine. Evidence-based medicine is with us for 30 years now, and so most doctors who graduated during this time or were trained may think that evidence-based is granted and is like an undisputable wisdom how we should practice medicine. But I just want to make sure that we understand that this is just a methodology that was developed based on population genetics. And so the idea of evidence-based medicine is to use randomized clinical trials to generate statistical evidence of the efficacy of certain therapies or interventions, regardless of the mechanism of action of these drugs or intervention or any a priori understanding of the functional any functional evidence. And that was highlighted with the story with the nail in the head, that in evidence-based medicine, you must ignore your a priori functional understanding of the mechanism of the disease, which basically would be the essence of precision oncology or precision medicine, when we simply try to use this understanding. Of course, the problem is that if we make decisions individually on the patient level, we lose the statistical power, and that's the great antagony we are trying to solve, and this talk about how we try to solve this controversy. So just one story in my career, which was extremely important for me, was the case of a 60-year-old lung cancer patient who never smoked and had multiple brain mets and lung metastasis in 2003. And that was the year when gefitinib, and a very potent small molecule inhibitor of the epidermal growth factor receptor, was tried in a large randomized clinical trial, the INTACT trial, where patients were randomized, lung cancer patients, in two arms, the chemotherapy arm and chemotherapy plus gefitinib arm. And this trial was a complete failure. So there was absolutely no statistical difference between the two arms. You can see that the lines are overlapping in the Kaplan-Meier curve. And that was really a shock to the oncology community at that time, because we thought that we can treat all lung cancer cases because 80% plus of patients express this protein, and we thought it's a very important growth factor receptor kinase that could, if we could inhibit, we could effectively treat all lung cancer patients. So, and the same year after these results were presented, we met this patient, and we did not only check the expression of the protein, but we also did a FISH analysis first, if fluorescence in situ hybridization, to look at the gene copy number, because our reasoning was that amplification of the gene in case of EGFR2, another family member of this family, worked well to when we had as a predictive diagnostic method to predict response to a trastuzumab, another targeted therapy approved in breast cancer. So we thought that maybe the same idea could work here. So we did first this amplification and detected a huge significant amplification in this gene. And then, but we also sequenced the gene. I had some prior experience how to do Sanger sequencing at that one time, and that was the only method that was available. We didn't have next generation sequencing in 2003. It was invented in 2005. So we had to do direct sequencing, and that was difficult in FFPE blocks. So we had to develop a new technology how to sequence short fragments that are only available in this type of samples. And we found a deletion, a large but in-frame deletion in the kinase domain of this tumor, and this was obviously amplified. So we reasoned that this mutation could be an oncogenic mutation because there was an advantage of the tumor from having this allele multiplied. So based on this, we initiated an investigator-initiated clinical trial and requested this drug to be used for this individual patient. But of course, we had a really hard time to convince the ethical committee to approve this individual trial, because we had the highest level of evidence that this drug was ineffective. So we just used biological reasoning to treat this individual. But finally, we got the permission, and of course, this was a transformative experience for me as a translational scientist to see how this patient remarkably responded to this treatment and with complete response, and she was tumor-free for 5 years and died of unrelated causes. So basically, we could prevent premature death from cancer just by using our understanding of the biology and the molecular pharmacology of a drug. Later, we used ex vivo experiments and preclinical evidence to really explore the mechanism. We showed that actually the mutant forms of this protein actually is much more sensitive to this inhibitor. So there is even a pharmacological selectivity towards these inhibitors. But then we had to wait until 2009 when finally the results of a randomized clinical trial, IPAS, was published, where patients were randomized in 2 arms, EGFR mutation positive and negative, and it was shown that EGFR mutated patients actually benefit more from the targeted therapy than from chemotherapy, in contrast to vice versa. Non-mutants benefited more from chemotherapy. Now, there's a lot of lessons from this early trial. That, of course, the aim was to use this 4-arm randomization to also control the possible prognostic effect of this genetic subtype of this tumor. And in fact, there is a positive prognostic significance of EGFR mutations, but still it's clearly shown that, of course, tumors driven by these oncogenes that are caused by this oncogene are responsive to this therapy. So now we call EGFR mutation a biomarker, but this is not really something invented by nature to provide us a biomarker. We are conceptually diagnosing the cause of cancer in these patients, the driver mechanism, and the nail. And we just use very logical reasoning to treat this patient. But still, based on evidence-based medicine, we needed this trial to get this drug approved. Now, today, it would be very challenging to get permission to do such trial, because it seems unethical on the one hand to treat a driver, a patient with an obvious driver with chemotherapy, or a patient with obviously lacking a driver with a targeted therapy instead of chemotherapy. So it's a scientific and ethical challenge as well. So if we go, but we were very enthusiastic at that time that actually we can combine the concept of precision oncology and evidence-based medicine by creating smarter groups of patients instead of using histology or tumor site as our target, because we understood that each driver mutation can cause multiple different type of tumors depending where that mutation occurs. It can transform into a tumor, a lung cancer cell or a breast cancer cell, but also on the other side of the coin, each cancer can be caused by multiple different mutations. And if you want to treat the patient based on the driver mechanism, the nail, we have to diagnose the exact cause why the patient has cancer. But so it may be better to group patients based on the presence of the target driver rather than histology, and that led later, of course, the rise or the appearance of tumor agnostic therapies we are aware of. And then by 2020, we reached an amazing milestone. The pan-cancer whole genome project that was published in 2020. And authors of this amazing work reported that they could identify the at least one driver mechanism in 95% of cancer patients. So it's amazing. We are now in the post-genomic era of cancer research. And according to Cosmic, the actual number of possible cancer-related driver genes is around 700. It's countable. So we all think about the human genome as an infinitely complicated thing, but we have to recognize that we have countable number of nucleotides in our genome, and we have countable number of genes, and there must be a countable limited number of cancer genes. So, and we are running out, so we don't really discover many more now. So it's saturated. So we should be already there. So we should be practicing precision oncology now. We have the NGS tests that can easily, for a few $100 of laboratory costs, sequence the gene set. So we can identify the cause of cancer in practically all of our patients. Now, the difficulty is if you go to the FDA's homepage to see which genes are approved as biomarkers, as companion diagnostics, there are only around 30 out of the 700. So it's a long way to go to theoretically use this information of the genome for therapeutic treatment decisions. And what we see is a mathematical problem of the long tail distribution of cancer genes, their mutations, and the combinations of these mutations. So at the beginning, we were focusing, of course, on the more frequent cancer genes that occur more than 1%. But most of the cancer genes and alteration occur in less than 1% of the population, that these are the majority of patients. And on the top of it, each patient on average carries a combination of 4 or 5 drivers. So even if we target 1 out of the 5, maybe the other 4 cause resistance, or maybe we should choose another one instead of the first one that could independently inhibited and cause cell death, or we must have a combination, but we also have to know which combination. And that's why we see on the market that many NGS test providers cover hundreds of genes, even whole genomes or whole exomes, because simply technologically it makes sense, because it's not more expensive to run a large panel really than a small panel. But majority of the molecular information is not used today, like 95%, and also still many reports are clinically not actionable, like 50% or more is not actionable, and because they don't have any FDA approved biomarker that can serve as a companion diagnostic of a targeted therapy. And we have so really to generate all the clinical trial based evidence to decide which therapy we should choose for our patients, I made this calculation that we would need 14.4 million years. Actually, if we use real-world evidence, that time can shorten significantly to 200,000 years. So it's still not good enough. And the other challenge is with this multidimensionality. Is just one example. Again, this is one of our patients. Again, a breast cancer patient who actually had a next-generation sequencing test that revealed that instead of each HER2 amplification, actually had an activating mutation. It occurs in 1% of breast cancer patients, but on the same time, the patient turned out to be a carrier of a BRCA mutation, which is more frequent in triple negative breast cancers, but it can occur in EGFR2 positives. So the problem is that we have statistical evidence that PARP inhibitors work in BRCA mutant breast cancer patients. We also have statistical evidence that HER2 inhibitors work in HER2 positive breast cancer patients, but we don't have any clinical trial-based evidence today how patients who carry BRCA mutations and positive for HER2 actually respond to these drugs, whether these alteration cause resistance or sensitivity to the other targeted therapy. So we will need a clinical trial that actually compares these two drugs on patients who have this combination of mutations. But of course, now we are really facing a very small population. And if I add that this patient also had another 3 alterations that are already known to interfere with the efficacy of HER2 inhibitors and PARP inhibitors, we can see the level of complexity we face every day in our clinical practice at our molecular tumor board. So because not most alterations are not approved by FDA as a biomarker, we follow guidelines created by our societies, ASCO, ESMO, how to evaluate the evidence that actually link an alteration to a possible treatment. somehow standardize our way of thinking. So these are the ESCAT scales and ASCO scales. But the problem is that we don't have really a consensus database, because by definition there is no consensus. And so when there was a study just published in this year in January in Molecular Diagnostic Therapies, they showed that when 154 biomarker drug pairs were presented to 14 specialists, there was deviation from consensus in 50% of rankings. So, and also we know that if we send the same molecular profile to 2 different molecular tumor boards in 2 different universities to ask for treatment recommendation from the board, we experience extremely low concordance rates, many cases below 50%. And this also corresponds to the outcome of these patients. And so what we see is that like 14 different publications reported outcome of patients treated with therapies chosen by molecular tumor boards that varied from 0% to 67%. So of course, if you were a patient, you would now wanted to know which cancer center to go. But of course, then we must ensure that the same scientists and physicians are part of the same tumor board for eternity, and they never go for holiday. So what could be the solution? And of course, you can guess that we need an outside-of-the-box solution. We need a new technology to be introduced, and yes, this is the AI thing, the computational solution. So we need a new class of devices that actually combine the strengths of and the advantages of evidence-based medicine and precision oncology. I think the solution is to have validated, personalized treatment planning machines that actually can be validated by clinical trials. But these machines or algorithms should enable us to choose the right targeted therapy for every cancer patient, but they should be deterministic and standardized and reproducible to democratize access to the most effective therapy based on evidence for our patients. So this is something we have been working on the past 10 years and many others, but this has been a long journey. We reviewed 10,000 cases on our molecular tumor board to really develop the right logic and the right chain of thoughts that maximizes our chances to make the right decision. And then we program this into a multidimensional knowledge graph that can be the backbone of a reasoning engine that can combine information between that link multiple drivers with multiple targets and multiple compounds in this three-dimensional model, and to reason out which is the most important driver, target, and compound that is supported by highest level of aggregated evidence of multiple sources to really imitate a reasoning of a molecular tumor board on a high complexity level. So using this technology, now we can not only link one genetic alteration to one therapy in pairs, but we can combine all what we know about all the totality of the alterations to the patients to rank treatment options. And with this technology, We can provide multidimensional decision support, but still in a way that is explainable, open box, and expected by human experts as the right chain of thoughts, and can be explored, we can explore, if we want, all the reasoning steps that led the treatment recommendation to really have control on the AI and eliminate the risk of any hallucination. And then, of course, the most important thing is whether it works or not. So it was really important for us to actually do what we envisioned and test the machine, the personalized treatment selection machine, or actually the DDA means digital drug assignment. This is the name of the algorithm. In a clinical trial. So we used the clinical trial data of SHIV-1. That was a very famous clinical trial conducted by Christophe Tourneau in Institut Curie. And because that was the really first randomized precision oncology trial where actually patients with predefined actionable targets were included, randomized, and then received the matching targeted therapies. And what we found after the reanalysis of these results, that the algorithm could really separate patients who really benefited from the matching therapy, and 4 times higher chances than those that received targeted matching therapy based on the presence of a single genetic alteration that were negatively predicted or predicted to fail by the model by simply computing in the totality of information of the tumor genomics. So with this, I think we are advancing to this dream to really practice personalized precision oncology by now using multidimensional clinical decision support system, and that is scalable and that can really enable us to use the totality of NHGS test results and other genomic information. And this year, when I talking to you, I already presented our results on the AACR on a large data sets generated on avatars, tumors treated in mouse, and to show that indeed tumors that receive targeted therapies that predicted by the model to be sensitive to a targeted therapy have statistically higher chance to respond to that targeted therapy than compared to chemotherapy. But those targeted therapies that match, even if they match a single driver, may be worse compared to chemotherapy if the model predicts resistance. So this is really exciting, and we are about to publish what we already showed at ASCO, that actually lung cancer patients who received in real world targeted therapies that were also supported by high dimensional model actually had an excellent long overall survival. 50% of patients actually alive after 5 years. That's multiple times longer than patients treated with chemotherapy or unmatched targeted therapies. But I think for a biological point of view, it's exciting to see that it is possible to outsmart cancer, because there is a limited number of genetic alterations that can occur, and combinations and mutations and resistance mechanisms also follow a biological algorithm, and there's a limitation of which type of combinations of mutations are advantageous for the cells, so most of them deleterious. So that is a countable number of strategies, molecular strategies that the tumor can use. So we can simply outsmart it. So now we have hundreds of targeted therapies, and we can sequence the whole genome, and we can use computational models, AI, to battle the complexity of cancer genomics and cancer biology. And so it's an extremely exciting time that starts now, and I'm looking forward to work all our colleagues and with you in the future, in this bright future, to really make sure that this journey will not take us 14 million years, but just few decades maximum, and finish this in our lifetime, to make sure that we integrate precision oncology in the clinical workflow using computational methods and AI. Thank you very much for watching this presentation, and I hope that I shared some thoughts that also help you in your career. And thank you very much for your attention. Thank you.